It is always fun to share some things with this group. I am going to get through a lot of information, but I'm not going to go into a lot of the details of the science. And the reason why is because this is a special time for the project I'm going to be talking about, the DIAGNOSE CTE research project. Is coming to an end after many, many years. And so the idea was today to just kind of share the history of this, what has been done, what are some of the key findings so far, just to let you know what we've been up to with this project. So here's some disclosures. And we're going to go back in time. 9 years ago, Barack Obama was president. Corona was a beer, and the big pandemic that year in 2014 was Ebola. Zoom really just meant something that was going wicked fast. Office buildings were full of people working because people didn't work from home. Billie Eilish, if those of you want to kind of go into that side of things, Billie Eilish was only 12 years old. Moving into what we do here in terms of things like neurodegenerative disease, the lymphatic clearance system was discovered only 2 years earlier. iPhone, the iPhone 6 and 6 Plus was released that year. And believe it or not, the ATN classification scheme, the first one, not the one that's being reviewed right now for kind of updating, that first ATN was still 2 years in the future. And so at that point, really, I think the understanding of CTE was still in its infancy. In fact, if you look at just the number of publications that had come out with chronic traumatic encephalopathy in keywords at that point in 2014, when we initially submitted this grant, there were only 135 publications to date at that point. Now, moving forward, between 2014 and now, there has been over 1,100 additional publications with a total of over 1,200. And believe it or not, yes, 87% have not come from Boston University. 9 years ago, the National Institutes of Neurological Disorders and Stroke released a request for applications for a U01 grant. And that was going to be funded. The $16 million was going to be funded by monies from the National Football League that were pledged to the foundation of NIH in order to fund one project to do what it says on the right. And that was the idea. And money is donated from outside groups, foundations, corporations, you name it, to be used for NIH research or NIH-funded research is supposed to be really held through with a firewall. The money is given to the foundation of NIH, and then there's supposed to be a firewall between FNIH and NIH and the peer review process and the decisions for funding. So September 30th, 2014, letters of intent were due. We submitted one. October 31 in 2014, the applications were due, and we submitted this grant for a multi-center project, expanding upon a previous R01 grant of mine called the DETECT study. And I was really, really fortunate that after begging and pleading with some incredible people, who I had been friends with and worked with, Drs. Jeff Cummings, Eric Ryman, and Martha Shenton, all agreed to be multiple principal investigators with me serving as the contact and overall lead PI. And so the way it was supposed to be was peer review was finished in the end of April. Our grant actually then received a great review and was the kind of best score by a large distance to the next grant that was reviewed. And the council at NINDS selected our grant to be the one grant to be funded. And the earliest starting date was supposed to be June 15th. And then came the long wait. We waited and waited from the time we were told that council selected our grant for several months with no word, actually no word. And then finally in September of 2015, so that entire summer, we were supposed to have gotten funding and started, but didn't hear anything. In September, there was an additional NINDS council meeting that revisited this decision and again selected our grant to be funded, deciding to fund it fully from the NIH budget, not using the NFL money that was given to the foundation at NIH. And then we received our notice of grant award on December 12th for a startup of December 15th. So I got to tell you, back 9 years ago, I was not a conspiracy theorist. I really wasn't. I didn't really know what conspiracy theorists were all about back then, but I became one when I turned into the center of a conspiracy. I'm not going to go into this in detail. I'm just going to show you some pictures that if you're interested, you can follow up on these things. But it led to a congressional investigation about the process for the decision-making that went on that summer for funding this grant. And the congressional investigation basically found that the NFL and its minions, many of whom were kind of senior scientists around the country, were doing something that wasn't that kosher, trying to change the decision about funding a grant solely for what seemed to be conflicted reasons. And they basically, the congressional report said that the NFL tried to influence unrestricted research gift, and it turned into a decision that had to go up to Francis Collins, who was the director of the NIH at that point, who just said, forget it, NFL, we don't want your money. We're going to find the funds to fully fund this grant to that group. And then a little known fact that this is my claim to fame. My colleague, Dr. McKee, was one in the Time 100 most important people in the world. Well, I didn't get that. I got some recognition in this way. The NFL's 100 most important people. Well, that year, Tom Brady was number 10. He didn't get up to number 1. People were a little upset. But guess what? This guy, Bob Stern, was number 7 most important person in the NFL.
So Bob, I always thought you were part of the deep state, just for the record. You know?
So yeah, we don't talk about that. So it was enough of that stuff. It was just so disconcerting. And so I stopped my deep state work and started to do the science, which is what it was all about. And so we had this grant. We called it a wonderful acronym, the DIAGNOSE CTE Research Project. You can see what the full name of it was. And it turned into a little more than $17 million. And instead of it being a 7-year study. It turned into an 8-year study. And now the end of funding is in 22 days and 4 hours and 40 minutes from now. So that's a long time. It was actually 10 years in the making. And now after 8 years of the actual funding, we're just about done. So yeah, as I mentioned, am the contact or lead principal investigator, but these are the folks who were so kind to agree to do this with me and have spent so much of their effort and thinking over these last several years, and it's been an incredible honor to work with these folks. To give you an idea of what people looked like back then, because this was 9 years ago, you can see I had a little bit more hair on top, less hair on the bottom. And let's see if there's people here who you know. You may recognize Dr. O and Dr. McLean. And here you can see that Mike Alasko, who was 11 years old at that time, was starting to grow a beard. And Brett Martin looked a whole lot younger. And our fearless leader around here at the time, Dr. Cowell, was the center director for the Alzheimer's Disease Center and much younger. Some of you know Dr. Cantu, and here's Jesse Mez, who I think was 13 years old at the time. And things have changed through the years. So we also have this external advisory board of incredible folks representing a variety of incredible input that they could provide to us. Chaired by Dave Knopman from the Mayo Clinic, retired Colonel Hack, who played a huge role in the Department of Defense work on traumatic brain injury. Brian Hainline, who's the Chief Medical Officer of the NCAA. Mike Haynes, who many of you know is one of the top former American football players ever in the Hall of Fame. Tom McAllister, he's now the retired chair of the Department of Psychiatry and one of the heads of the CARE Consortium. Arthur Toga, who runs the LONI at University of Southern California, and Mike Weiner, who, as many of you know, has been the longstanding principal investigator of ADNI. So what was this study? How did we design it to kind of hopefully work? Well, the idea was to make it interdisciplinary in a way that could break down all kinds of silos, institutional, specialty, degrees, you name it, breaking down all the silos so we could work together across those disciplines. We had, I think it's 13 different institutions involved. Here is a list of the people who've been involved with this project around the country, actually around the world, including folks in Gothenburg, Sweden. And it's been an incredible group of people who have played a huge role and made a gigantic impact in this. So the specific aims for this project was first to collect and analyze neuroimaging and fluid biomarkers for the in vivo detection of CTE, and then to also characterize the clinical presentation of CTE, examine the progression of CTE over a 3-year period, to refine and validate diagnostic criteria for the in vivo or the clinical diagnosis of chronic traumatic encephalopathy, to investigate genetic and head impact exposure risk factors for CTE. Very important to us was to share project data with researchers across the country and abroad in order for other people to have access to what we thought would be a very important database. The organizational structure of this project has been, I think, key to making it work. We have this administrative team of the 4 PIs. We have a publications committee that's chaired by Jeff Cummings, one of the PIs. And then we also have a coordinating center that has played the kind of central role of the orchestration of the entire project. And I'll describe that a little bit more later. Then we had teams for all of the various areas of both data collection, the science, the analyses, including a risk team led by Rhoda O, with exposure science being led by Dr. Mike McLean, and the genetics led by Jesse Metz. We had a fluid biomarkers team run by Elaine Peskind at the University of Washington and the VA Puget Sound in Seattle, along with some great colleagues of hers, including Jeff Iliff, and then also folks at the University of Gothenburg, including Kai Blenau, Henrik Zetterberg, and Nick Ashton. We had a neuroimaging team co-led by 2 of the PIs, Eric Ryman and Marty Shenton, with Eric running the PET sub-team and Marty running the MR sub-team. We had a clinical outcomes team with Jeff Cummings and me as co-leaders, and Jeff overseeing the neuropsychiatric side of things, Mike Alasko overseeing the neurocognitive, neuropsychological side, and Chuck Adler from the Mayo Clinic overseeing the neurological and motor side of things. We eventually, as I'll mention in a little bit, developed a diversity, equity, and inclusion team overseen by Robert Turner at George Washington University. We had a diagnostic criteria team where I led it with sub-teams of the consensus conferences that I'll describe, overseen by moi, and new NINDS consensus diagnostic criteria for traumatic encephalopathy syndrome, overseen by Doug Katz here from BU. Then we had a data team with Yorgos Tripodis from the School of Public Health, and many of you know, as the overall leader, with Joe Palmisano as the data management sub-team leader, and Yorgos overseeing biostatistics. And then we had a lot of input from folks over at NINDS, because this is a UO1, which is a collaborative project, which means NIH and the investigators work together to make it happen. And then we would get input from our advisory board and then provide them with information about how it's going. So if anyone wants to get a much more detailed description of the design, as well as some kind of summary data of who our participants were, they can go to this paper led by Mike Alasko, which was a methodology paper for the project that came out a few years ago. So who did we study? We recruited over 240 participants, but the goal was then to have a solid set of participants equaling 240. These were all men between the ages of 45 and 74 years old. 3 groups that were based on history of exposure to repetitive head impacts. So we have kind of a high exposure group, a medium exposure group, and an unexposed group. The high exposed group were 120 former NFL players. And again, all between 45 and 74, and they weren't recruited based on symptoms. It was open to anyone who met the rest of the inclusion and exclusion criteria, but the idea was to get a spectrum of symptom severity, from no symptoms to some mild symptoms to dementia. Same thing for our former college group. So we enrolled 60 former college football players who never went on and played any other contact sport and never went beyond the college level in football. And again, same age and the same spectrum of symptoms. And then we had this unexposed control group. They were screened to be asymptomatic based on telephone screening. They couldn't have had any exposure to repetitive blows to the head through sports, through a traumatic brain injury, or even a mild traumatic brain injury or concussion, couldn't have had combat military experience, and again, all asymptomatic. Everyone was then seen at one of these 4 performance sites, which meant flying people. Most people had to be flown from wherever they lived around the country to one of these 4 sites, where they underwent a 3-day baseline evaluation. And you can see who the site principal investigators were. In Arizona at the Mayo Clinic in Scottsdale, Chuck Adler was still is the site principal investigator. And then PET scans for the folks who went to Arizona were done at Banner Alzheimer's Institute. In Las Vegas, our site was the Cleveland Clinic, Lou Rubo Center for Brain Health, and Charlie Bernick was the site PI there. In New York, NYU Langone with Laura Balser as the site PI. And here in Boston at our place, I was the site PI with our MRIs done, not here, but at the Brigham and Women's Hospital. So what did people have done during this baseline evaluation? We collected a whole lot of data for things like repetitive head impact exposure history, clinical data of all different types, and all participants were examined and completed questionnaires and you name it to get all this type of clinical information. So let's go over the specific aims and show what we did and some of our key findings. And so let's start with the collection of neuroimaging and fluid biomarkers. So in addition to those clinical exams, everyone had a lumbar puncture for CSF, a whole lot of blood collected. We collected saliva. We haven't begun to do anything with that, but all of our biofluids were then processed locally, put on dry ice, and sent to Seattle, where our freezers are and our biobank is. And so we got hundreds and hundreds and hundreds and thousands, actually, of aliquots of CSF and various blood products and saliva there. Everyone also had a pretty extensive MRI with several sequences, including volumetrics, diffusion, resting state fMRI, magnetic resonance spectroscopy. And then people had 2 different PET scans, one an amyloid PET with florbetapir, and another for tau PET using the fluorotaucipir radiotracer. So here's one of those wonderful things about timing. We completed all 240 of our 3-day follow-up evaluations on February 26th of 2020. For those of you who can remember this kind of weird thing that happened around the end of February, beginning of March, that COVID thing struck. but we were able to have completed our 240th 3-day evaluation right before lockdown. And I'm not going to go into detail about the findings, but I wanted to just show you some representative findings of our biomarker data so far. This was a paper that just came out around a month ago in Alzheimer's Research and Therapy, where we looked at amyloid PET to see if former American football players with cognitive impairment and dementia had Alzheimer's disease as the cause of their impairments, because the florbetapir PET is FDA approved for the purpose of ruling out Alzheimer's disease as the underlying neuropathological disease causing the person's impairment. And lo and behold, what we found was that no, our folks did not have elevated florbetapir findings. And even the ones who had dementia, they did not have elevated florbetapir. And in fact, the numbers were much lower than one would have expected just based on the kind of data that are out there for community folks with and without symptoms. So this is one thing that could have some both scientific as well as clinical and medical legal type of ramifications to show that these former football players don't seem to have Alzheimer's disease as the underlying cause of their impairments. So then Michael Osko did a really cool job looking at in vivo fluortaucipir PET, that's the tau PET tracer, and postmortem neuropathology in 6 of our participants who had died after their baseline evaluation, and whose brains were then donated and examined by Dr. McKee and her team. And what we found was that fluortaucipir PET was pretty good, but only for high-stage CTE pathology. And the specificity of this tracer, fluortaucipir for CTE p-tau, still is uncertain. There's off-target fluortaucipir binding in both the hippocampus and thalamus that makes it difficult to interpret some of our strongest findings. And that we still need a lot of work ahead of us to especially try to understand the best type of PET tracer for CTE tau. In other words, it doesn't exist yet, but at least for high stage CTE, this tracer does seem to do a pretty good job of detecting the p-tau pathology. But then what about the larger sample? How did it seem to do? And so Yi Su from Banner Alzheimer's Institute has led the efforts for this. This is under review right now. Revision is under review at Alzheimer's and Dementia, looking at the results of a fluortaucipir SUVR. This just gives a little taste of it in terms of statistical brain maps. This was trying to replicate in a way and expand upon a former paper of ours in the New England Journal of Medicine a few years ago. With this much larger sample, more kind of appropriate to make some inferences from, our DIAGNOSE sample showed that fluortaucipir did have some pretty significant differences in binding between the former NFL players and controls, between the former college players and controls. And still a little bit of difference between the former NFL players and the folks with more moderate or medium amount of exposure to the repetitive hits. Michael Osko has been overseeing some great work on white matter hyperintensities and white matter abnormalities as associated with CTE and in people with a history of repetitive head impacts. And in this paper, he found that indeed there was a relationship between, in this case, the age of first exposure and the age of first exposure to playing football and the total amount of white matter hyperintensities, but only in our sample that was on our older side, the ones who were age 60 and above, such that the folks who started to play earlier in life, pretty much as you can see, earlier than, let's say, age 12, had the highest amount of white matter hyperintensities. Monica Lee is here now at BU. She was doing her postdoc when she started this project at UCSD. She worked with Mike and colleagues and looked at the association of vascular risk factors in CSF and the imaging biomarkers with white matter hyperintensities, and found that these risk factors, these kind of vascular risk factors, and some biological correlates of white matter hyperintensities differed between the former players exposed to repetitive head impacts and our asymptomatic unexposed men, and that vascular health, p-tau 181 seen in CSF, and diffusion tensor imaging indices of white matter integrity showed the strongest associations with white matter hyperintensities in the former players. Then we have a few different MR studies. This one says draft under review by authors. I believe it's been submitted. This one says submitted soon. It has been submitted to Nature Communications. I'm not going to go into the details now. Then we have some papers looking at our fluid biomarkers. This is a paper by one of our fellows here, who's back in Amsterdam, completing her PhD, Suzanne van Emmerongen, and she looked at inflammatory biomarkers for the neuropsychiatric syndrome associated with CTE, neurobehavioral dysregulation. Okay. What about the clinical presentation of CTE? What have we done for that? We gave so many different types of tests, neurological exams, symptom validity measures, neuropsychological tests of attention, executive function, learning and memory, language, visual spatial functioning, emotional facial perception, then measures of dementia severity or functional independence, measures of sleep, olfaction, pain, substance use histories, health status questionnaires, and that turns into a ton of data. We also collected a huge number of questionnaires, measures of neuropsychiatric status in terms of affective lability, behavior, depression, anxiety, apathy, loneliness. I won't go into really any details here, but Mike led this paper for summary of the neuropsychological test performance of our former players. We have a paper that is hopefully in the final stages of review, characterizing this neurobehavioral dysregulation, seeing that there seems to be it seems to be a valid construct, a neuropsychiatric construct that is associated with this history of repetitive head impacts, and it seems to have a few different kind of subconstructs of explosive behavior, being impulsive, having emotional discontrol, and affective lability. Additional representative findings. This one says under review. It's actually accepted for publication. Where we're looking at Parkinsonism in our former players. And this one is also under review in neurology clinical practice, looking at clinical outcomes and tau pathology, but specifically looking at the impact of sleep apnea, and some interesting findings there. Then we were supposed to examine the progression of CTE over a 3-year period. That was our aim. And so we had planned to bring back all of our participants for another three-day in-person evaluation, 3 years after their original baseline evaluation. That was going to be fantastic. And then that COVID thing hit. And like so many things going on, we had to pivot, change our plans. And instead of a 3-year evaluation, we changed it to a 4-year follow-up, but all remotely. And so we got updates on all the history variables over telephone and Zoom interviews. We did neuropsychological tests done either by telephone or through Zoom using kind of modifications of some standard neuropsychological tests, but done via Zoom, and also added online computerized testing with the CANTAB measures. But all the rest was the same for the neuropsychiatric and questionnaires and the study partner questionnaires. I forgot to mention everyone had to have a study partner. Those were all done using REDCap online at baseline, and again, the same way at follow-up. But obviously, we couldn't do things like MRIs and PET scans remotely, but we were able to get in-home venipuncture. So we could get blood from our follow-up evaluations that could then be processed locally, aliquoted with kits that we would provide to the group of examiners, phlebotomists around the country, and it would then be sent on dry ice to our freezers at the VA Puget Sound. So we just finished our last follow-up evaluation just last week, thanks to the incredible group of folks at our coordinating center here at BU. We were able to get a pretty good follow-up retention. So this is the total number that could have had a follow-up, 230. There were 240 total, remember, but 10 people have already passed away, and so the maximum number we could get would be 230. We have done 208 follow-up evaluations. 10 people formally withdrew. They didn't want to play anymore. And 12 people, no matter how hard we sleuthed to try to find them and have them get back in touch with us, 12 people were just lost to follow-up. But that means that out of 230, we had a 90% retention rate, which is pretty darn good. For our 4-year follow-ups, you can see that we had over 90% success in getting all parts of our remote 4-year follow-up assessment, including that in-home blood draw. 98% of our follow-up participants agreed to and underwent an in-home blood draw. So now we have both baseline and follow-up blood aliquots in 204 people. The next specific aim was to refine and validate diagnostic criteria. Because of time, I'm not going to get into the real weeds of this, but part of what this grant did was to oversee, coordinate, and run the first national Institute of Neurological Disorders and Stroke consensus workshop to define the diagnostic criteria for traumatic encephalopathy syndrome, or TES. This was held in Phoenix like 4.5 years ago. And we had this unbelievable group of people who participated, including folks from NINDS as observers, including Walter Koroshetz, who was the director of the Institute. We then had some 20 panelists, with some who were unaffiliated with our actual data collection or the study. They weren't investigators for the DIAGNOSE CTE research project. Some of these names you might notice or know, folks like Ron Peterson, Ross Safant from Mass General, Barry Jordan, Kristen Dams-O'Connor, and Dave Brody. Then we had all these voting panelists from this study, including our own folks in red, and then some investigators in the project who were non-voting observers, including Dave Notman and here from BU, Yorgos Tripodis. The goals of that consensus workshop were to develop evidence-informed expert consensus criteria for TES, which is viewed as the clinical disorder associated with neuropathologically diagnosed CTE, and to improve upon previously proposed research diagnostic criteria for TES by trying to maintain the sensitivity that existed, but increase the specificity of the previous criteria for the specificity for detecting underlying CT pathology. And then to develop a criteria structure that could be amenable to future updates without having to start all over again. Doug Katz, our very own Doug Katz from BU, oversaw this modified Delphi process to achieve consensus. The panelists all agreed to how this Delphi process would work. There'd be a maximum of 4 rounds of voting. It would all be anonymous voting. And then there'd be revisions of wherever we were with the consensus criteria after each of the voting rounds. And there would need to be 80% or more agreement amongst the panelists for anything to be considered a consensus. So we began with previously published provisional criteria for TES, something we did back in 2014. And then we used new literature, old literature, and new evidence for clinical pathological correlation, including a paper that Jesse Mez oversaw from the UNITE study, where the original 2014 traumatic encephalopathy syndrome criteria were examined in terms of its sensitivity and specificity for actual CTE. And you can see within this blue square, the sensitivity was great, 97, but the specificity is really lousy at 21. So we went through 4 rounds of voting, revisions, and about 2 years later, there was consensus, and we published this paper that if you're interested in the details of it, I recommend going to that. Very general kind of summary of it is that it's a stepwise process to come up with a diagnosis of TES. People would need to have a substantial exposure to repetitive head impacts in order to have the diagnosis. They'd need to meet core clinical features of cognitive impairment, specific types, and neurobehavioral dysregulation, or both. There'd also have to be a progressive course. If all that was met, the criteria require that if whatever the clinical presentation is not fully accounted for by other disorders. And if all those three things are met, the person would be considered to have TES, and then there would be the determination of the level of functional dependence using some specifics in the criteria. So first comes TES as step one. And then we'd like to be able to get to what does that mean for CT pathology? So we developed levels of certainty for CT pathology using some specifics of the type of exposure, the amount of exposure, some of the specifics of the level of functional impairment, and everyone needing to have cognitive impairment, but not just neurobehavioral dysregulation, plus a whole bunch of supportive features that I'm not going to get into now. And that would lead to levels of certainty for CT pathology. And if you were to look at the following algorithm without my warning you that it could be a little daunting, you'd probably scream. And here's the algorithm that is actually not that complicated. I'd recommend looking at the paper if you want to find out more. The future, however, is the use of biomarkers, because the current TES criteria do not have inclusion of biomarkers to be able to come up with the level of certainty for CTE, because the panelists all agreed at that point 2 or 3 years ago that we weren't ready. There were no good biomarkers yet for CTE. So remember this, remember that everyone had all these things done, but then we also held multidisciplinary diagnostic consensus conferences twice monthly over video conferencing. Even before Zoom existed, we would be meeting over video conferences. Around 15 clinicians have been part of this panel doing these multidisciplinary consensus conferences for all of our 240 participants. We'd have a case presentation. There'd be real-time red cap voting independently. There'd then be discussion, and a final consensus diagnosis would be reached, with everyone having, as part of their diagnosis, use of the NINDS consensus diagnostic criteria. This is just a kind of reiteration of what I just said. But you can't imagine that this same group of investigators from all around the country met every 2 weeks. We'd have to have a quorum of a minimum number of people representing different sites, different specialties. They met every 2 weeks for 5 to 6 years. Incredible dedication of this the amazing group of folks. And they completed around 450 diagnoses using our NINDS consensus criteria. Then comes investigation of genetic and head impact exposure risk factors. I'm going to speed through this stuff. As many of you know, we collect a whole lot of data in our various studies about head impact exposure histories and TBIs and concussions, and all that is used to create a cumulative head impact index that I won't go over in depth. This is a paper led by Dan Danishvar using UNITE data to kind of validate a new version of the cumulative cumulative head impact index. But we also compare the professional football players with the college football players. We look at years of playing football, the age of first exposure, et cetera. Genetics are an important part of what we're looking at. And starting with something pretty easy to try to get a grasp on, and that was the ApoE4 genotype. Preliminary findings that have not been published yet, so I'm not going to go into this at all, really, is that yes, indeed, there is an association between E4 being an E4 carrier and having reduced volumes in the frontal cortices, but only in our older players. And it's not associated with tau deposition or clinical performance. Very importantly, Jesse Mez and colleagues have been looking at MAPT haplotypes and subhaplotypes, and those data are going to be analyzed soon. After we started, however, we decided that it's really important to look at additional very critical variables, and that is to look at the impact of racial disparities and social determinants of health. And this really started after the horrible murders in 2020. And so we created this diversity, equity, and inclusion team that Robert Turner has been leading. We've developed a variety of additional measures to be able to improve our ability to understand things like early childhood, social determinants of health. And then finally, the last specific aim was to share project data. And so you can imagine all the data. We have around 7,000 variables just for baseline data per participant. Then we have all of the additional variables for follow-up. So we're talking about like 10,000 variables per participant coming from all over the place. So that takes a huge amount of data management and coordination, et cetera. And thank goodness we've had this incredible data team led by Joe Palmisano at BDAC in our School of Public Health here, along with Brett Martin and a group of fabulous folks who many people in the Alzheimer's Disease Research Center also know. So all of our baseline data are just about to be available for qualified investigators through the FITBR data sharing repository. But that's just kind of a subgroup of the data. And so we were determined to have our own data sharing portal that would allow investigators from all over to be able to have all of the rich data that we have obtained. And so that's going to be released through a new website, the DIAGNOSE-CTE-data.org. You can't go onto it yet. It's still under construction, but it's going to be opened up and released by the end of the month. So the question is, are we there yet? You might be questioning, are we at the end of the talk yet? The bigger question is, can we diagnose CTE during life? Are we there yet for a clinical diagnosis? Well, we're much closer due to the incredible participants that we have had and their study partners. We're much closer because of the great collaborators in this study, as well as our staff and trainees. But there's still a lot that we don't know. We still have a lot of data that we can analyze to try to help, but many critical questions remain, like what clinical features are specifically related to the p-tau pathology of CTE? Is it are clinical changes associated with just that initial sulcal depth p-tau deposition, or Do we only see things once there's later medial temporal lobe involvement? We just don't know if it's that, or maybe it's p-tau related neurodegeneration. So we have to figure out the specific relationship between clinical features and what part of the CTE pathology is going on. But also some of the clinical features may be due to non-p-tau related pathology or from repetitive head impact exposure or to other causes. Is there a relationship with inflammatory responses from the initial RHI exposure or later neurodegeneration? Are there chronic and progressive changes from a single moderate to severe TBI that a lot of these folks have? What are we looking at when we're talking about white matter damage? Is it arteriosclerosis? Is it rarefaction? What about comorbid diseases and conditions? We know that there's a whole lot of comorbidity going on. Do those things play a role with the clinical manifestation? And are the clinical features associated with just other factors separate from RHI exposure, including just idiopathic psychiatric disorders, sleep disorders, chronic pain, mental health outcomes from psychosocial factors related to retiring from sport that these former professional and college athletes were participating in, and then very important racial and associated inequities in social and cultural determinants of health. I'm going to skip a couple things. So what's next? We are actually submitting a grant very soon for DIAGNOSE CTE. Michael Lasko is leading this. We've got some great collaborations with Alzheimer's disease research centers around the country to take this the next step. And we can't go into the details of it because the grant is not yet submitted. Moving forward, though, I want to leave you with this idea that there does still need to be a lot of research. We still need to close those knowledge gaps a little further, but how do we determine when we've had enough data to make some important changes before we make changes to clinical practice, and very importantly, before we do something to change some of the activities that lead to these later life very important problems? It's a complex issue, a really complex issue. One that I can't get into in the 1 minute I have left. But once we can diagnose CTE during life, we'll be able to begin clinical trials for treatment. And if we can detect it early, which I think we're going to be able to do through, in particular, some new blood tests. If we can detect it early in the disease course prior to symptoms, just like in Alzheimer's disease, we'll be able to conduct clinical trials for disease-modifying medications that might be able to delay or prevent the symptoms from happening. And if we can understand those specific risk factors a little bit better and resiliency factors for CT, maybe prevention strategies can actually be implemented. And if that doesn't work, perhaps we could just learn from our animal friends. It says on the bottom, if you can't read it, I read that story about dementia and former NFL players. Maybe we should reconsider this. So special thanks to everyone involved with this study, especially my BU CT Center investigators, staff, trainees, everyone across the DIAGNOSE CTE Research Project, our funders, and most importantly, all of the research participants, study partners, and now brain donors and their family. This is that coordinating center group of amazing people here at BU, and a lot of you are in this somewhat old picture at this time. Anyways, that's it. Thank you very much.